Hi, my name is Sebastián Iturbe Piña. I'm going to talk about microRNA in Littorina littorea. This is part of the course of useless biology of the genomic science program of UNAM Juriquilla. We are going to talk about the article name microRNA regulation in extreme environments, differential expression of microRNAs in the interdidial snail Littorina littorea during extended periods of freezing and anoxia, by Cal K. Bígaro, Samantha F. Kornfeld, Julia Mastrovsky, and Kenneth B. Story. Okay, so in the introduction to Littorina littorea is also known as the common periwinkle, is a resilient marine snail inhabiting interdidial zones. Its ability to survive extreme environment conditions makes it a fascinating subject of study. Littorea is a marine gastropod mollusk that thrives in rocky shores. Its strong shell and robust physiological mechanisms allow it to withstand harsh conditions, making an important species in coastal ecosystems. This species is commonly found in the interdidial zones of North America and Europe. Its habitat is characterized by varying tidal fluctuations, which significantly influence its survival and behavior. Littorea faces numerous environmental challenges, including anoxia and freezing temperatures. These stresses occur due to daily tidal changes and seasonal temperature fluctuations, impacting its metabolic process. Okay, but what are microRNAs? MicroRNAs, also called mRNAs, are small non-coding RNA molecules that play a vital role in post-transcriptional regulation of gene expressions. They are crucial for various biological processes, including development, cell differentiation, and stress responses. MicroRNAs are short, approximately 22 nucleotides long, non-coding RNA sequences that regulate gene expressions by binding to complementary messenger RNA targets. This binding inhibits translation or promotes degradation, thus controlling gene activity post-transcriptionally. MicroRNAs are involved in numerous biological functions such as development, apoptosis, and responses to stressors. Their regulations impact key processes like cell cycle progression, differentiation, and metabolic processes essential for the organism's survival. Emerging evidence highlights that microRNAs are crucial for the adaptation of organisms to environmental stresses such as freezing and anoxia. They modulate cellular pathways that facilitate survival under harsh conditions by regulating genes associated with stress responses. So, in the article, this outlines the techniques utilized to investigate the role of microRNAs in the stress responses of Littorina littorea. The study highlights the selection of specific microRNAs and the approaches taken to measure their expression levels. Okay, the study employs stem loop reverse transcription PCR as the primary technique to analyze microRNA expression. This method is highly sensitive and allows for the amplification and quantification of specific microRNAs, ensuring accurate measurements even in low abundance samples. The selected microRNAs for the study included those that are highly conserved across species similar to Littorina littorea. Particularly, they focus on microRNAs 1A1, microRNA 34A, and microRNA 29B. These microRNAs have been implicated in metabolic regulation and stress responses, providing insight into the roles in the snail's adaptation mechanism. The expression levels of the selected microRNAs were measured in response to freezing and anoxian conditions. The study revealed significant upregulation of these microRNAs in different tissues, indicating their crucial role in mediating stress responses during challenging environmental conditions. In food muscle, microRNAs such as microRNA 1A1 and microRNA 34A exhibited increased expression following freezing and anoxia, while microRNA 29B also demonstrated significant upregulation. This response suggests that these microRNAs may play crucial roles in modulating metabolic rate and stress responses within muscle cells during hard conditions. In hepatopancreas tissue, microRNAs 1A1, microRNA 34A, and microRNA 29B were notable upregulated during freezing, while microRNAs 210 significantly increased in response to agnosia. These findings highlight the involvement of specific microRNAs in regulating metabolic pathways that are crucial for energy conservation during stressful events. These findings indicate that microRNAs may aid in the survival of Littorina littorea by regulating protein synthesis and facilitating metabolic adjustment under extreme conditions. The ability of microRNAs to inhibit messenger RNA's translation allows these snails to conserve energy and adapt to fluctuating environmental stressors. The conclusion: the research emphasizes that microRNAs are critical mediators in the response of Littorina littorea to environmental stresses such as freezing and anoxia. By supporting translation suppression and enhancing antioxidant defenses, microRNAs play a vital role in the pro-survival mechanism of this organism, offering insights into molecular adaptations in invertebrates. Okay, but what is next for microRNAs? In this article, they measure the microRNAs only on freezing environment or only in an oxygen environment. So a good question could be how the microRNA regulates in both freezing and an oxygen environments combined.